Whether you realize it or not, you are constantly encountering a plethora of microbes. In addition to this, no matter how careful you are, your cells are getting damaged all over your body, from scraped knees to an intense workout to just brushing your teeth. When you add in the surveillance for mutated cells and just recycling cells that have lived out their life, you'll start to see that our immune systems are putting in a lot of hours. Hello and welcome back to the Science Scene. My name is Shelby Bradford and I'm the Geeky Goth. I'm a PhD candidate in immunology and microbiology, and I'm sharing my love of this subject with you to help make some sense of and maybe gain an appreciation for this complex madness that is our immune system. We're exploring this immune system piece by piece, and it's time to jump back into another immuno adventure. If you want to follow along, make sure to like and subscribe so that you don't miss any immunology lessons. Now, normally you probably connect the immune system with things that give us immunity. That's definitely an important element, however, it's hardly the whole picture. Most of the list we just went through doesn't even involve any memory. But you hopefully agree they're all pretty important items to take care of, and preferably quickly. So how does the immune system pull off this trick? Of course, the easy answer is the innate immune system, but we're here to dig deeper. How do these cells and proteins keep us in tip-top shape? What do they actually do? Let's start with a quick review. We can mostly lump our innate immune cells into two groups, phagocytes and granulocytes. Phagocytes' claim to fame is their ability to gobble things up, while granulocytes are responsible for gushing out a collection of chemicals and proteins, which together we call granules, all over the place. How are these immune functions though? Let's break it down. The process of eating another cell or debris is called phagocytosis. It might sound ridiculous, eating things as a defense, but remember, our cells don't have hands to pick up the trash. And when this junk is outside of the blood vessel, it won't go through the liver to get filtered out. So we need some way to clean things up, or everything will look like frat houses in bad movies about college. Phagocytes do this cleaning up by reaching out, yeah, cells aren't always little blobby dudes, and enveloping their target to pull them into their cell body. Now they've got this trash bubble in them. With the help of a lot of other molecules, they maneuver this trash bubble to essentially the cell garbage disposal, where a variety of enzymes and acids are going to break down this item into pieces. These pieces will either be recycled into the cell to make other cell things, or used to activate other parts of the immune system. So this process helps to not only tidy things up, but also assists other immune functions. Next, granulocytes. We saw our granulocytes take center stage in our last video, where we discussed many of their molecules promoted inflammation. This helped to prepare our body for an immune response by activating and recruiting immune cells and making us a less hospitable home for invaders. But another function of some of these granules is to directly attack cells. These may be invading microbes like bacteria, fungi, or parasites, but it could be our own cells too if they're infected. Some of these granules are going to punch holes in cells, which, besides causing the cells to spill their insides out, also lets in other granules to attack. Other molecules are going to help induce apoptosis, that special programmed cell death to remove compromised cells. So innate immune cells have some pretty hardcore functions, but how do they know when to use them? Remember, cells don't have eyes to spot pathogens or affected cells. Instead, they more or less rely on touch, kind of poking things. Our immune cells are decorated in a variety of proteins that are collectively called pattern recognition receptors. These will identify general features, proteins or lipids or other chemicals found on microbes, because these receptors can latch onto these structures almost like puzzle pieces. They also use similar receptors to spot chemicals produced by our cells when there is some internal threat. When something binds a PRR, it begins an inflammatory cascade, and then it's off to the races for our immune response. With all of that covered, let's jump in and talk some more about these innate immune cells and what they do. Neutrophils, if you remember, kind of have a foot in both camps of granulocytes and phagocytes. So they are both gobblers and gushers. However, I hinted before that these cells have another trick up their sleeve. If things start getting too out of hand, neutrophils will undergo their own special cell death. Unlike apoptosis, which was supposed to be cleaner because our cells collapsed in on themselves into little blebs and in this way reduced inflammation, when neutrophils self-destruct, one of the goals is to induce inflammation. It does this by releasing its DNA. Because this is a molecule supposed to be on the inside of cells, seeing it on the outside of cells is a sign of danger. However, this mass of DNA also serves to capture microbes, making them easier to grab onto and destroy. This feature dubbed this process neutrophil extracellular traps, often shortened oh so perfectly to nets. That is when cells go boom. On to another dual functioning cell, the macrophage. Macrophages exist to eat. However, while filling up on their pathogen or debris treat du jour, they'll also be responding to the environment around them. Pro-inflammatory molecules will encourage them to respond with inflammatory molecules of their own, driving immune stimulation. As immune responses progress though, either suppressive cytokines or an increase in markers of cell damage on other nearby cells will be recognized by macrophages through other special receptors and will shift these macrophages to more suppressive functions, where they'll now aid in promoting more tissue repair activities. Our final phagocyte is the dendritic cell. It also gobbles up pathogens and debris like our macrophages. However, its goal is not to stay in place and aid in the fight on the front line. Instead, it hangs on to these fragments from phagocytosis, which will be used as antigens, and they move to lymph nodes to assist in activating adaptive cells. Dendritic cells will then create special cytokines to further stimulate these adaptive cell functions. We'll return to this process when we visit adaptive immune cells in a future video. On to our granulocytes. Mast cells, basophils, and eosinophils are all potent granulocytes. And we saw in the last video how inflammation could be triggered by their granules. However, each often has a special niche. Mast cells predominantly survey our tissues, which is why they were our initiating cell in our inflammation video. Basophils, meanwhile, are primarily scouting our blood for invaders, while eosinophils are also more tissue-dwelling, particularly lymph tissues and the GI. All of these cells have been implicated in various allergic responses, but also, especially for the case of eosinophils, in defense against parasites. Lastly, our innate oddball, the natural killer cell. While they don't fit into the granulocyte or phagocyte group, they are uniquely poised to destroy infected or compromised cells. But how do they know who to target? By checking IDs. All cells with nuclei, that place where the DNA is, have a special molecule on their surface, dubbed Major Histocompatibility Complex Class 1. Look, I don't name this stuff. It regularly grabs bits of protein from inside of the cell and shows it off, like a child with a new drawing. All of this is indication the cell is healthy. However, when cells are infected or damaged, they'll often have too little MHC class I on their surface. Alternatively, these compromised cells may display an abundance of other special receptors that signify this compromised status. This is the NK cell's cue to activate. They can either engage with proteins on these cells to induce that self-destruct sequence apoptosis, or they can bombard the cell with deadly compounds. While brutal, this is a fail-safe to prevent further damage from infection or injury. Our individual cells are made to be replaced. So that is our exploration into the cells of the innate immune system. They may be a bit erratic and sometimes messy, but they do a good job of keeping most things under control. Sometimes though, things do get out of hand and they need backup. That's the time for the adaptive immune system to step in. We'll talk about that side of the immune system and more when we return for more immuno adventures and more awesome science.